Well, I feel extremely honoured to have been asked to give this lecture by AWISE and Somerville, and in conjunction with the museum where Dorothy worked. This multifaceted woman has really fascinated me once I've started to read about her. And you can see her here looking at some electron density, and you're going to say, I don't know what electron density is, and I'm going to switch this on so it works. And also in her lab looking at some crystals down the microscope. So she was an amazing woman, and the more I read about her, the more I'm amazed at the number of different aspects of life in which she shone. Oh, sorry. And yesterday was in the 100th International Women's Day, and she featured as one of the 100 women who during the last 100 years, lots of hundreds here, had changed the world in the UK. So it was British women who changed the world. And this was what the small thumbnail of her in The Independent on Sunday said. Also, I don't know how many of you heard Points of View by Lisa Jardine at 10 to 9, quite a lot of you, in which she bemoaned the fact that art and science seem to have become separated and rather polarised, and recalled the 1951 Festival of Britain Crystallographers Club that got together to design some amazing wallpapers and things that were up during that festival, and I'll show you one of those in a minute. So her list of honours is really incredible, and is hidden here, also a member of 14 foreign academies, at least one on each continent, but not Antarctica. I couldn't find Antarctica. She was Chancellor of Bristol University. She was President of Pugwash for 13 years, reluctantly but highly effectively. And she was also the President of the International Union of Crystallography for 3 years, the first woman president, and we have the second currently President of the International Union of Crystallography. There are many words written about her. This is by Georgina Ferry, who I'm happy to say is with us today, and this is the Fellows of the Royal Society biography by Guy Dodson, which is very illuminating as well. So there's many buildings named after her. These are the out of Oxford buildings that I could track down. Keele, York, where Eleanor and Guy Dodson, who worked with her in Oxford, moved, and Dorothy visited many times when they were there. And then since she was Chancellor of Bristol University, there's a big building there. And in Oxford, we have 94 Woodstock Road, which is where her home was. And I took this yesterday, and guess what? They were digging up all along here, and I photoshopped out the bollards. And I heard that there was a movement to put a blue plaque on this building, but I actually couldn't find it or get close enough. There's a Dorothy Hodgkin quad in Somerville, and I don't know if any of you who walk past this every single day know where that is, but it's quite near here. Yes, some people do know where it is. Okay, so you can wander around the science area and look for Dorothy Hodgkin Road. She solved the structures of, and I will explain what this means in a minute, cholesterol iodide, one of the family of cholesterols that you all know about for your health, penicillin, the precursor to all the antibiotics we rely on now, vitamin B12, and you can see that the number of atoms, and I hope everybody is up to speed with an atom, because we're going to build them from there. We can't start from zero, but you're made of atoms, if that helps. Insulin, which was her tour de force, that has 829 non-hydrogen atoms. But more importantly than these particular structures, her work paved the way for a huge number, as you'll see now, over 50,000 three-dimensional structures of biological molecules, from which we get much information that we can apply to drug design, to pathways in human health that both go right and wrong. And this is an incredible little quote from the end of her Nature 1934 article, which I'll come back to in a minute. At this stage, such ideas are merely speculative, which is that you could get the structure of a protein from a crystal. But now that a crystalline protein has been made to give X-ray photographs, it's clear that we have the means of checking them, and by examining the structure of all crystalline proteins, arriving at far more detailed conclusions about protein structure than previous physical or chemical methods have been able to give. This gives us two questions to answer immediately. What's a crystal and what are proteins? And I hope that I'm about to answer both of those for those of you who haven't been brainwashed already by these. I wanted to sort of put you into the Dorothy Hodgkin sort of mindset. There was no email, and this changes your life. It's much nicer probably because you're not being hassled all the time by it. But this was the hot news in LMB Cambridge when insulin was solved. This MFP is Max Perutz, and this was the big news on the bulletin board on the 3rd of August 1969. So no email. So this is what I hope to cover in the lecture. That was just a precursor. What is protein crystallography? If I fail to answer that, the 5 people here from my 1972 leaving class of school will not forgive me. Why do we do it? What's the point of it? Then I'm going to take you through a crystallographic experiment, how Dorothy would have done it and how we do it now and what has changed. And then perhaps a little glimpse into the future, but end with some words about Dorothy Hodgkin. Before I do that, I want to tell you what my father said to me when he heard I was about to start lecturing in Oxford. My father was an electrical engineer who was a REME in the war, trained to be a cleric when he came out of the war, and 3 years into his curatorship he went blind in a week. And he was an amazing man, as those of you here who met him will know. And this is what he said to me. A lecture is an event where information passes from the notes of the lecturer to the notes of the student without going through the brains of either. This is his 80th birthday picnic up in the hills of Northumberland. I recently modified this for... So here we go. What is a protein? Well, many of the functions in your bodies are governed by these proteins, and that's why we're interested in them to do with health, to understand the cell cycle that's implicated in cancer, to understand how plants grow, just how nature works, really. They're made of nitrogen, carbon, oxygen, and hydrogen atoms. There are 20 different ones in nature, but they're all of exactly the same scaffold. So there's a nitrogen, a carbon with an oxygen, hydrogen, and then this R group here can be 20 different things as in nature. It could be just a hydrogen atom, it could be a carbon and 3 hydrogens, etc. So this is called the side group, the side chain of this. What they do, these amino acids, is that they form long chains. And these chains are very, very reminiscent of chains of beads. So here's a chain of beads, kindly made for me when she was 4 by my daughter here. And it has several elephants and teddy bears and things in it, but you can see that the beads are different colors. And the 20, I'm not sure there are 20 different colors, but if you imagine these amino acids forming chains, they never form Y-shapes because it's very hard to invent a molecular machine which can actually produce a Y-shaped molecule from DNA to RNA to a protein. We'll come back to that machine in a minute. One of these beads, one of these amino acids, this one's a blue fish, is absolutely tiny. So if you can imagine a millimeter on a ruler and take a millionth of it, that's how big this amino acid is. So what the chemists can do through modern sequencing techniques is identify that at position 6 we have today orange rhinoceros, right? We've got, say, a glycine. We've got a particular amino acid we can identify. They can tell us what number is at each position on here. What they, as yet, are unable to predict or do, but are working on it, is how this thing is going to wrap up like wet spaghetti in some horrible shape like this in order to be the protein that's going around in your body and doing its operation. So these chains can become extremely long. So the one I'm going to tell you about in a minute is about 400 amino acids long. And these are the 20 amino acids. So they're all exactly the same scaffold, COO, which is the red ones. The blue ones are nitrogen, and the grey ones here are carbon, and 2 of them have sulfur in, which we use to great advantage in our crystallographic studies. So here's our snake of amino acids. We don't know what the 3-dimensional shape of that looks like. All we know is the identity of these different amino acids at different positions. This is tumor necrosis factor, which is actually the body's way of trying to give remission from cancer tumors. If we look at how tumor necrosis factor actually folds up, we see that even in your worst nightmares, you would never be able to imagine that this is what it did. What this is plotting on the left-hand side is just the string through these beads. So what we've done is strip off all these side chains and bits sticking out, the Christmas tree branches, if you like. We've stripped them all off, and we're just showing the connectivity of this protein going around like this. If we put all the atoms on, and we imagine atoms as little round balls, then this is what it looks like. And this tells us nothing, except that we've got a surface. It might tell us what the channels and so on are, but it doesn't tell us what's connected to what. So we rather annoyingly tend to picture them like this, when really they're not space, they're solid. 75% of that volume is filled with atoms. And then this one goes around in threes. So here it is, 3 of them, different colors, and there we see the ribbon diagram of 3 of them. These are disulfide bonds at the top, holding the thing together, bridging it. If we look at Dorothy's molecules, penicillin is shown here, and again, a convention which is slightly annoying when you first meet it is that at the junctions there's a carbon atom, but we know that they're there, so we never put them there. So, so all the joins there are actually a hidden carbon atom. This is vitamin B12. So this is 25 atoms. We're going up to, I think I said 60. And then here we've got 929 atoms. And you can see the different renditions of these. Here we've got all the side chains on. So we've got all the beads, the rhinoceroses, horns, etc. are still connected. Here we've taken the string, and we show one of the motifs that we find a lot of as an alpha helix here. And we've got another one, which you'll see in a minute, called a beta sheet. And here it is atomistically, and you'll see that actually this isn't a very useful way, usually, for us to render it. If we go to the big molecules, the Nobel Prize for Chemistry last year was awarded to Venki Ramakrishnan from the LMB in Cambridge, MRC LMB, Tom Stites from Yale, and Ada Yonath from the Weizmann Institute, for this absolutely enormous molecule. So you remember I said a minute ago that insulin had 829 non-hydrogen atoms. This one has 293,658 non-hydrogen atoms. Absolutely incredible achievement. The central dogma of our production of proteins in our body for digesting our food, moving our muscles, etc., is that DNA gets transcribed to RNA, messenger RNA, and then gets translated by this molecular machine, the ribosome, to the proteins that you need for your body. And I've got one here kindly donated to me when she retired by Margaret Adams, which is lactate dehydrogenase, which is a protein in your muscle that when you run up and down the stairs very, very fast, turns over to give you that energy, and then you get a pain in your leg because you went too fast, and that's the lactic acid, which is a product of this enzyme giving you a pain because you overdid it. Now, this actually, don't laugh too hard, but this is from a dogfish. So this is dogfish lactate dehydrogenase. And before any of you say that dogfish can't run up and down stairs, they do need to flap their tails, so they actually need this too. You can see this again is the string through the beads that we have a look at here. So if we now, so we've answered the question, what's a protein? The next question we ask is, what's a crystal? So this is a parade of soldiers outside Buckingham Palace. And what I want you to imagine is you've got a parade of soldiers here, lines of them, and that we're going to put another layer on the top, all standing like this, completely the same, and just touching the next one, but they're all identical. And then we're going to make that an infinite array or semi-infinite array, and we're going to pile many layers of soldiers up. And then we're going to put them in a swimming pool. And each of these soldiers is equivalent to a protein molecule. And now we're going to get about 100 cannons, and we're going to shoot the cannons at the soldiers, and the cannonballs are going to bounce off. And they're going to form piles on the other side of this array of soldiers. And we're going to look at those piles and say, right, I've got 6 in this pile, so I've bounced off something quite hard in this soldier. I've got none in this pile, or 1, so that's something quite small on the soldier, like his ear. But then occasionally a cannonball is going to lodge in the soldier array and get stuck there, lose its energy, and knock off either a foot or an arm or a... and this is the problem of radiation damage in protein crystallography. So now... because we can't get all the photons that we shoot at these crystals. So what I'm going to do is get a very intense beam of X-rays and shoot it at a crystal made up of biological molecules, but the interstitial space between those molecules is going to be water or solvent. It's not going to be air. And this is going to give us a lot of trouble. And Dorothy Hodgkin was the first with Bernal. Bernal did an initial experiment, but Dorothy followed it up systematically to discover that actually if you didn't allow these crystals to dry out, then all the soldiers would stand in rows. But if you took the soldiers out of the swimming pool, they would all just go in different orientations. And what we want is an ordered array of biological molecules with them all in exactly the same orientation, so that we'll get the same reflection angle of these cannonballs bouncing off them for the shape that they are. Now, crystals come in all sorts of shapes and sizes. So I found the biggest crystal in the world known to man, and this is 37.4 feet. And before you tell me that's not an SI unit, it sounded much more impressive than 11 meters somehow. Here's a man, right? And here are the crystals. I mean, they're absolutely incredible. This cave is at 112 degrees Fahrenheit, though. It's not a comfy place for tourists. And, you know, the request to air condition it, it has been pointed out that all the crystals will disintegrate if that's done. So you have to be pretty devoted to go and see them. I would love to do that one day. By contrast, a protein crystal, this is a calcium sulfate, selenite, a form of calcium sulfate. The protein crystal, here's one that's 0.2 of a millimeter. So take an inch and divide it into 100, and that's the size of that crystal, so pretty small. Now, they're very fragile. So those of you who have worked near me will know that, you know, we get this. It looked lovely before I opened the drop up. Then they all dried out and they were no good anymore. And the challenge, it's a huge challenge to grow biological molecules into these so-called crystals, which are really a quasi state of matter between liquid and solid. Here's Dorothy Hodgkin looking down the microscope, because even her crystals were of a size where she had to use a microscope to see them. And we use smaller and smaller crystals, and we're happy with them. So there are many variables in crystal growing. One of them is crystal fingers, but there is concentration, pH, additives, temperature, grandmother's maiden name, gravity. And some people can grow crystals and some people can't. And we have robots to set up 3,000 conditions with a new protein, and then we try to grow and get crystals. And if we get one, we say, great, and we take it off and try to hit it with X-rays. So we grow crystals by putting our protein liquid in with something like ammonium sulfate or some chemical, many different ones we can try, in a drop here. So we've diluted the protein by a factor of 2 because we've added a drop of another liquid. And then we have a reservoir here of the liquid at full concentration. It's at half here. And very slowly, water vapor will come out of here to try and equilibrate the whole system. We seal it up. We use grease to seal it up. So a greasy job. And then we hope, we go and we pray to whatever God we believe in, and we hope that a crystal will grow, either fast or slow, and it'll be ordered. The variables, one of the interesting variables is gravity, and I'm just going to take a small diversion and tell you about when we explored that dimension of crystal growth. The theory is that if you've got a crystal growing in a drop, you are actually going to deplete the area around the crystal because the molecules have already joined the crystal. So therefore, you've got lower density material right next to it. Lower density material will rise to the top if you're on Earth, and it'll set up a convection current going around like this, which will then stop further molecules lying down on the crystal face. So the theory is, if you send this into space to grow there, you won't have this problem. You get big crystals. So we did that. This is reverse transcriptase from HIV, which I worked with David Stammers, who's I'm happy to say is here on this project, and he went off to watch the launch but left me in the lab at home. Now I was not long an ex-nuclear physicist. I'd only moved 1 year over from nuclear physics to biophysics, and unfortunately the press rang up and they got me, and because all the other guys were, it was a 2-week delay in the launch, and they had a great time over there till it went up. So Nicholas Schoon for the Daily Telegraph, don't read this, don't worry about trying to read this, but basically it's a fantastically accurate article saying that we're sending part of HIV up into space, trying to grow better crystals so we can design better inhibitors. AZT and or zidovine both bind to this particular enzyme, reverse transcriptase, and it says that we're hoping to get better crystals. But the next day, to my horror, I had a phone call from the press office who said, what had I been telling the press? Because this had appeared in a paper I don't normally read, which is a Daily Star. And basically it says madness. I mean, the quality of the typeface is about the same as the quality of the journalism. But it says madness, boffins want to put AIDS in space. Is there something about scientists that makes you wonder whether their Bunsen burners are on full pressure? It goes on to tell you, and as I say, it's hard to read, so don't worry about it, but that, of course, there is a nuclear-powered Russian satellite out of control coming down. There's an AIDS-carrying STS going up, and it implies, without actually saying it, that they're going to meet and spread AIDS and nuclear waste all around the globe. So this was my first brush with the press with protein crystallography, and as many of you know, there have been quite a lot since. So these are crystals of a neuraminidase from bird flu. This is a noddy tern. It wasn't this noddy tern, but a noddy tern provided the original influenza virus, because birds, as you all know, also get flu, but it's a stomach disease. These were grown on Earth, and the ones that went up on Mir, because it turns out that STS wasn't up long enough for our crystals to grow. But if you can get them on Mir with the Russians, then they're up there for 3 months, and you've got more chance. These ones were only, the Mir ones were absolutely marginally tiny bit better, and really not worth the enormous cost. So at that point, our love affair with space ceased. So why do we bother to find the three-dimensional structures of biological molecules? What can they tell us, and how can they help human health? Well, Dorothy's first two here, penicillin, that's probably obvious to all of you, that was the precursor to modern antibiotics. To understand their shape and how they interact and how they act as antibiotics enables us to design and better ones, and now, unfortunately, to try and design ones that will not give, or new ones to which we're not resistant. Her work on insulin underlies the whole idea that you can design a better synthetic insulin or a recombinant insulin that would mean that diabetics don't have to actually inject so often, because you can modify it such that it's absorbed more slowly. So this is a very clear indicator of the utility or the use of these structures. There's a huge reason as well to do them to understand pathways. Even if we don't understand exactly how they work, we can use the structures to do experiments to determine molecular pathways and which proteins are involved in which functions. And then the third one I've already mentioned is influenza neuraminidase. This three-dimensional structure of that has allowed the design of Relenza and Tamiflu anti-flu drugs. This is insulin. It's got a zinc atom in the middle of it, and it forms together. This is actually a hexamer of it, which is Dorothy Hodgkin and the Dodson's structure. And here is a flu virus, and the neuraminidase is this black mushroom-shaped protein on the outside. By cleaving that, the size of this, by the way, we're getting a bit bigger. We're now 1/millionth of a centimetre across rather than 1/millionth of a millimetre. So we're getting bigger. And in wild waterfowl, you can find 9 types of this, and that's the hemagglutinin, the H. This is the N of H5N1 and H1N1. There's 16 of these. And by chopping that off, it can be crystallized, and you can get the three-dimensional structure. And these are different subtypes of the 9 neuraminidase. These crystals are good crystals. This one's a human one. This is from an English duck, Ukrainian duck, and a noddy tern, just to get it international here. These ones are no good because the soldiers have gone a bit crazy here, and they're not staying in a nice regular array. There's a few in an array, and then another array at a different angle, and they would not get through their passing out parade like this. So these are no good for our experiments. This is what influenza neuraminidase looks like, looking down on it and to the side of it. And this is what it looks like if you look at its accessible surface for a drug. And you can identify by soaking in the thing that it chops off on the outside of your cells, It's called Mr. Blobby. Actually, it's called sialic acid, but it looks like Mr. Blobby there. This is the difference between an experiment I did with the sialic acid and one without the sialic acid, and I can see where it binds. So I can now identify where this is, the active site of this enzyme, and 3 Australians actually designed a drug that would fit in there, which was Relenza, and they designed it by looking at the electron density, which is what we get from our experiments, and saying, oh look, there's a little hole there, and if we made something that looked like the normal thing on your cells, sialic acid, and we fill in this little hole here with an extra few atoms which will bond to this part of the protein, we might be able to make a drug that, if you take it or sniff it, would block you to the normal thing on your cells. So it's a bit like putting a key in a lock, and then another key comes along and you can't get it in because there's already something in there. And in that way, you can perhaps disable flu viruses from multiplying too much. You have to take this drug very early on, otherwise it's too late and you're already infected. And this is a sort of structural biologist picture of what's going on here. This is Relenza bound in the noddy turn neuraminidase. This bit's of the protein, and this is the drug here. And this is a model of what we build onto our electron density. So now I'm going to give you Avatar 2, and you're going to go on a little journey, so all hold on, into the active site of the neuraminidase from this one's English duck. So this is one that we solved in my group, and off we go. So if anyone's feeling sick, just leave. Here it's coming for you. So there's the tetramer of neuraminidase going round, English duck neuraminidase. So that's the head from that little black stick I showed you on the outside of the flu virus. It's rotating. These represent beta sheets, and that's what it looks like in the atomistic view that I showed you, the space-filling view. And here we're going into the Relenza binding site. Whoa! And this is the drug bound in the active, the second binding site actually on that. This is the electron density. So the blue chicken wire is what we actually see in our experiment, and then we build in what we know the drug looks like. So, and then we're going to go out again. And I thank Ed Lowe very much for making that for me. The second binding site you can see on the tetramer there. Bye-bye. Off it goes. So, the experiment for Dorothy Hodgkin and for now. I hope you're convinced that this is a worthwhile escapade, our mission to find three-dimensional structures. So what I'm going to take you through is the X-ray source, and here's the X-ray source at the ESRF. I'll show you the local one in a minute. In Grenoble, it's at the confluence of 2 rivers, and there's a lot of trouble because it's alluvial silt and it sort of sunk when they built it, and they had to underpin it quite a lot. This is our crystal, and I'll come into why it's in this funny loop in a minute. This is the so-called diffraction pattern. These black dots are the piles of cannonballs on the other side of the soldiers. So they have bounced off the biological molecules and they've collected together in heaps, and they are, this is a big detector, a reusable electronic film, and we get these images and we can do some mathematics on it and get some extra information that we need to do some other experiments to do, and we can get to the electron density around the atoms, and then we can build a model into that. So at first the model doesn't look so good. I mean, this is not very convincing from the things I've shown you, but we have techniques to clean this up, and so we get beautiful electron density and makes you believe in atoms. And then you can build in your amino acid sequences that you know because you've got them from here. Okay, so the basic experiment is that we need an X-ray source. Dorothy Hodgkin had a lab source, an X-ray tube, or we now use synchrotrons. We have some optics to condition that beam and point it at where we want to put it. Then we have the crystal, which is totally out of proportion here, as you probably realize, just so you can see it. And then we have a detector. Now, Dorothy had 2 sorts of detector. She had photographic film, which she had to expose, go into the darkroom, develop, hold up to the light, have a look at. If it was any use, then she had to scan it and digitize it, or even just look at it and at first do hand measurements from it. And that's how, when I joined the LMB in 1987, we took data at Daresbury, the synchrotron, the UK synchrotron. We now have electronic detectors. We have two sorts. We have image plates, which we use in the lab, reuse electronic film effectively, and we have charge-coupled devices, which you have in your cameras and have been adapted for X-ray crystallography to give us large area detectors. This is fantastic because we can collect the whole pattern at once. Dorothy's second sort of detector was one which went along and just could look at one reflection at a time and had to be moved to where you would predict that next reflection, that next black dot would come. And it was very, very time consuming. It would take 3 weeks to get a data set. So the sizes we're dealing with now, we would look at a 50 micron, that's 0.05 of a millimeter, and 0.005 of a millimeter at the synchrotron. So we're getting smaller and smaller. We need better and better microscopes. This is a fantastic photograph of Dorothys Lab in the museum, and it's been taken by somebody who's on a ladder because that was the only way they could get far enough away. So it's been taken by Dennis Riley of this student, Harry Carlisle, and this is the X-ray kit. And can you see there it says danger? You probably can't see, but this was the only warning that, you know, there were X-rays about. And this is Dorothy working, I think, above, but I'm not quite sure. I couldn't identify the rooms in the museum at her desk. And now we believe in radiation shelters. This would, sorry, I meant to point out, this would take, as I said, 3 weeks for a data set. And it was totally unshielded, so you could put your finger in the beam if you wanted to, if you were stupid enough to. We now have a radiation shelter. This is a famous Myrtle, which some of you have used, and it took 16 hours for a data set. And this totally revolutionized what we could do in LMB when we got an electronic large area detector instead of having film that we had to process, and also single reflection detectors. Now we have Diamond. It looks like a snail, actually, there. And that gives us 10 million times the X-rays per second than Dorothy would have had. So just unbelievable extra photon flux. We can use much smaller crystals. It's a very parallel beam, and we can also change the wavelength. We can change the energy of the photons to suit our particular experiment. This is now to move on to actually consider how we're going to get the crystal into the X-ray beam. So on our X-ray experiment here, we've actually now, we're at the point where we're going to put the crystal in the beam. And Bernal and Dorothy Hodgkin realized, well, Bernal first, and then, as I said, Dorothy did the careful experiments about it, that if you actually put the crystal in a glass capillary tube instead of in the air, and kept it wet while you were taking the photographs, then you could actually get data from it. So it says that when they took it out on exposure to air, the X-ray photographs taken of the crystals in the usual way showed nothing but a vague blackening, so no spots. If you put it in a capillary tube of Lindemann gas, the first photograph taken in this way showed we were dealing with an unaltered crystal. So this was a fantastic discovery because that's what opened up the doors for our biological measurements. What you observe, though, at room temperature is a loss of diffraction. So this is a beautiful piles of cannonballs here extending way out to the edge of this detector, which gives us the best detail that we can. The best, if you like, atomic microscope on that protein. But after a few exposures, especially at the synchrotron, you find that the spots out here have disappeared and we're losing the detail that we need in order to find out exactly where the atoms are positioned. So if you look at the intensity of X-ray sources here from 1900 through, Dorothy was working in this period here, then we got rotating anodes, and now we have synchrotrons, which gives us this number here. And I'm going to tell you what this is, and then I'll be accused for jargon here, but this is an amazing unit. I love it. It's the photons, so that's the X-rays, number of X-rays, per second, per square millimeter, per square milliradian per 0.1% bandwidth. So that's not something you want to be examined on. It's the average spectral brightness, but it's basically the amount of zap, the amount of cannonballs you can put on your crystal, which has gone up hugely. Here is the Grenoble one again. When that came online in 1995, we all went off there with our capillary-mounted crystals, and we discovered they lasted a few minutes and not enough time to get any data. So at that point, people were already working on techniques to actually cool crystals while you irradiated them. In 1970, Michael Rossmann and David Haas had published this experiment, which basically they had a room temperature crystal and they plotted the intensity of the pile of cannonballs, or the reflection, with time as it was at the beginning and then as it was after a certain time into the experiment. And after about 100 hours, they had no cannonballs left. They were getting no scattering because they knocked off so many soldiers' feet, arms, ears, heads, etc., that they didn't have the right order anymore to get the reflections. However, if they cooled it to 198 Kelvin, which is minus 80 centigrade, then it lasted 400 hours. But nothing was really done about this for about 17 years, and then the protein crystallographers learned from the smaller molecule crystallographers the techniques of how to cool crystals. But they were very difficult to deal with because small molecule biological molecules can be brought out into the air, whereas the biological ones couldn't. And there was ice, and I tried for about 3 years to get these techniques to work from small molecule crystallography. This shows what happens to the capillary mounted crystal in the X-ray beam from a synchrotron. It only lasts a few seconds, and you get this discoloration due to the radiation damage because you're destroying the crystal order. So in 1990, a landmark paper of at least 1 3/4 pages was published in Journal of Applied Crystallography, which suggested that a good way of mounting protein crystals was to take a metal loop, dip it into the liquid in which it grew, pull it out, and then plunge it into liquid nitrogen at 77 Kelvin. Now, there was some technical difficulties with that. Number one, the metal loop diffracted like anything. So you put it up in front of the X-ray beam and you got lots of reflections. However, Catherine's hair, she was a baby at the time, was brilliant for this technique because fibers don't diffract very well. And when she saw me coming, she'd go like this with her hands because she knew I wanted a root to get the to make a loop. Also, Mike Glaser and John Cozier developed a wonderful cryostat, which was in the Clarendon lab, and that made providing a cool supply at 100 Kelvin, minus 173 centigrade, to your crystal possible and easy. This is a fiber, and she's probably sick of me saying this, but it's from Louise Johnson's mohair jumper, and it's excellent diffraction properties, hardly any background, when we made loops from that to mount our crystals. So these are quite small, it's about 1 millimeter across. So we started to use this technique to good effect, and we could get around the radiation damage problems largely. So this is a cryo-cooled Great Britain on the 9th of January this year. It's a wonderful satellite picture of the whole of the UK covered in snow with cloud cover. We don't like the snow in these experiments, and we have to be very careful how we cool these protein crystals, because remember I told you the soldiers were in a swimming pool. Well, if you freeze a swimming pool, it expands. So all the spaces between the protein molecules expand when you put them at 100 Kelvin, and that puts all the soldiers out of joint and they're not lined up anymore. So you have to add antifreeze and do various things to make this possible. But it has got us round a big problem with the synchrotrons in that it gives you a time factor of about 70 extra to take your data. So you get 70 times longer lifetime at 100 Kelvin than you do at room temperature, and you get better data quality and so on. So now most crystallographic data is taken at 100 Kelvin. And the red ones are the warm ones, the room temperature ones of all the experiments reported in one of our crystallographic journals. Now you may say to me, but I am not walking around the planet at 100 Kelvin, minus 173 centigrade. I'm at 37 centigrade, and this is not physiological. What we found, though, is that these structures are actually good to their they're the same at room temperature and at 37 as they are at 100 Kelvin. It's only a few details that are missing. So we can now send our crystals in a liquid nitrogen dewar to synchrotrons around the world, and they can arrive intact, and we can do FedEx crystallography. So that's this was FedEx, and this is one of my crystals that was sent to Stanford in 1998. Now, this may or may not work. What we find now with diamond and with ESRF, very intense X-ray beams, we find in fact that even at 100 Kelvin it's not good enough, and this may or may not work. So this was a crystal that I irradiated with Ed Mitchell at the synchrotron and then allowed to warm up, and you can see that something catastrophic happened. Gas was given off, it sort of broke up, and something very nasty is happening. And everything that was formed at 100 Kelvin is then released as soon as it gets warmed up. So we started to see radiation damage at cryotemperatures. And we also saw biological artifacts caused by particular damage to amino acids. This is probably a bit technical, but this is a limit that my group worked out for biological material. How many photons can you give it before you've spoiled the biological information in there? And the answer is we can give it an enormous dose of 43 million gray. A gray is a joule per kilogram. And just to put you in the picture, 3 gray would kill a hamster, right? So if you irradiate, you wouldn't, but if you irradiate a hamster, then you 3 gray would do it in. And we're now complaining that our crystals don't like 43 million gray. So this is our current problem. The main pathway, the main damage is done by the radiolysis of water, which is split up. And these are the reactions. These OHs we don't think are mobile at 100 Kelvin, but these are mobile. Okay, more seriously, we see particular damage to amino acids. This is a disulfide bond. This is our data with our model subtracted. And this tells us that we don't have the atoms we should there because that's red. Red is bad, green is okay. So now we're worried about this. And our big concern is the rate of damage versus the diffraction intensity. If I have a tiny crystal and I bash it with lots and lots of cannonballs, I get some information, but it doesn't last very long. If I don't hit it with as many cannonballs, I get more information, but it's weaker information. Okay, so what else is different from Dorothy to us? Computing. And I was totally stunned to be given a desk at Brooker AXS in Madison in September, and on my desk was sitting this, which was used, a bit of the computer that was used to refine the B12 structure with Ken Trueblood from UCLA and Dorothy Hodgkin. And there's a wonderful, in one of the books that I was looking at, there's a wonderful dialogue between them, all by post, of course, about doing this structure. So this now was twice the size of the laptop from which I'm doing this, and on which I'm not going to lift it up because it'll probably die if I do that. But I mean, this is really extraordinary, and they're all valves that we use this. Now the computer can predict where the spots can be, should be. We tell it the information, and it's a sort of video game where you try to match these little electronic predictions of where the piles of cannonballs are going to be. So I'm just now coming to an end, which is a good thing because I'm running out of time and everybody wants to get to the dinosaurs, to talk about how we represent these structures. So here, with the very kind help of the staff at the biochemistry department, I brought over the Lysozyme model, and there's a picture of Dorothy Hodgkin looking at it with David Phillips here. People used to have to build these when they'd solved their structure. Now, or they made balsa wood models, as is seen here. Now we have the computers, and we can do all sorts of fancy things. We can morph between structures. This is fibronectin, Staphylococcus aureus, the one that's turned into MRSA now in hospitals. It's part of that. And we got 2 different crystal structures, and it's morphing between the 2. And you know, the computer power we now have at our fingertips compared with having to build models like this and then carry them round. You know, you're giving a lecture and it takes 5 people to set you up. This is going to be one of the 100 objects. Some of you may have heard 100 objects on Radio 4, and it's Dorothy Hodgkin's penicillin map. And Yvonne Jones lent me these from the Tumour Necrosis Factor project when I first joined the lab. So you used to have to print them out on celluloid, and you can see that there's electron density in there. Then line them up and see, ooh, can I see a feature? Do I think there's a beta sheet through there or an alpha helix? Or I mean, just unbelievable that they managed to solve structures like that. It's very, very impressive. Lisa Jardine mentioned this wallpaper, so I thought I'd dig it off the web. This was actually, here's Dorothy looking at the insulin picture, and this was inspired that wallpaper, which was very popular. Okay, nearly done. So what's changed in the last 50 years? So the things that have really changed are that we can now express any old protein we like, or almost, by conning other organisms to do that for us. So bacterial, yeast, or insect cells, instead of going and getting 2 tons of spinach and pounding it up, or getting ox hearts from the abattoirs we used to do every Monday morning in LMB because you wanted a protein from them. You can now actually grow what you want up by conning another organism to overproduce it for you. We've got synchrotron radiation. This NP means that these factors were mentioned in the Nobel Prize statement of scientific background last year as being important for the ribosome project to enable the very small and difficult crystals to be looked at. We can make derivatives for getting the phasing, which I haven't talked about, but it's the extra information we need in our experiments. We've got crystallization robots that I mentioned. We've got X-ray detectors rather than single reflection detectors. They're very large, they're very accurate. We can cryocool our crystals to in some ways mitigate radiation damage. We'd like to do more, but we get a factor of 70 or so increase. And we get this fantastic computing power at our disposal, more powerful algorithms in the crystallographic software. So we see the number of protein structures growing exponentially. This isn't Moore's law, this is now the protein databank. This is open source, anyone can access these. This is 2010, we haven't really got going this year yet. Actually, this was February, I'm afraid it's not completely up to date. But this is really incredibly growing very, very fast. And journals now insist that you deposit your structures before you can publish. So what might happen in the future? Well, at the moment, the structural genomics initiatives are trying to take an entire genome and then find the structure of every protein that's coded for by that genome, even if they don't know what the function of those proteins is. And I think a big thing is to find out what they all do. It's a bit like stamp collecting when you have a structure, but you don't know actually what it does. We need to get going with membrane protein crystallography. It's very, very difficult, and I know some people in the audience are engaged with that. It's really challenging, and the main reason it's so challenging is when you go out in the rain, you don't dissolve because your membrane proteins are not actually soluble. But everything I told you about growing crystals relied on them being soluble. So then it's hard to actually crystallize them in the first place. We've got perhaps single particle imaging with free electron lasers will give us information. Bioinformatics, enormously powerful algorithms now being applied, and perhaps, this is one of my new colleagues in the Doctoral Training Centre, perhaps protein structure prediction may eventually obviate any need for crystals or X-rays eventually, but it certainly can't do that yet. As has been mentioned, Dorothy was part of the British Crystallographic Association, and in fact she was the founding vice president of it, and our remit is to promote crystallography because it's a vital part of many different parts of industry, chemistry, physics, biology, and this is actually Dorothy Hodgkin's original signature in the front of this. I'm now the proud keeper of this book for 3 years, so that's a crystallographic. And Dorothy also taught some very important people in her life, and so she had some very famous students, and I actually tried to bring this one up to date in 1989. I'm not sure how successful I was. She doesn't look very happy about it. I would like to thank my group, past and present. A lot of the things I've sort of mentioned in passing have been done in my group. My current group, Arij, and James and Robin, who are here, Enrique, who's in Mexico, Robert, who's gone to the consulting business, and Karthik, who's now in Berlin, Ed Lowe, who I've worked with for many years in LMB, who's our facilities manager and on whom I rely enormously, and then these collaborators, particularly the ESRF, for allowing us to actually do radiation damage studies with the beam, and even though it did not result in Science and Nature publications, but they gave us beam time to do that. I'd also like to thank Guy Dodson, Judith Howard, Georgina Ferry and Margaret Adams for providing me with some of the pictures in this lecture. All the students and staff at the Doctoral Training Centre, my new job, for their friendliness and welcoming me, and my colleagues and undergraduates at the Department of Biochemistry as well for having had me there for many years. Brasenose College, who've just let me in, and John, Ron, Danny, Jose and Kirsten for moving this morning, Tony, Chris and Kristin at the University Museum. And lastly, but by no means least, before I quote Dorothy, I would like to thank my family, particularly my husband, John Barnett, for his help with getting this lecture together, my 3 daughters and our 2 sons-in-law and my grandson here. for just being. So here's Dorothy as Chancellor of Bristol University, and what she said to the students on her last occasion, and I say to you too, for her, and I hope that some of you will live modestly and do serious things. And from the obituary by Guy Dodson, she's left an example of generosity and seriousness, which through countless friends, associates and students, is a legacy for science and for human affairs. And I'll end there, but I'd like to put this up for you to read because this is a fantastic quote from Max Perutz. And the main thing, I'm just going to read you the first two sentences. Dorothy was more Christian in word and deed than many believers I have known. She radiated love for chemistry, her family, her friends, her students, her crystals, and her college. So this is a really great epitaph, which as scientists we should all seek to emulate.